Good afternoon, everyone, and thank you for joining us today for our event, Widening the Search for New Dementia Treatments. I'm Katie Bray, and I'm Involvement and Engagement Manager here at Alzheimer's Research UK. And it is my pleasure to welcome you to today's event, in which we'll discuss research working to find new treatments for dementia. We've had a slight change of lineup, and so I'll be joined by Dr. Emma Murphy, who is stepping in for Dr. Emma Mead, and also be joined by Dr. Joyce Huang. But before we get into the discussion, I'd just go over some housekeeping. So during the event today, you're welcome to switch on the subtitles using the CC button at the bottom of your screen. These subtitles are generated automatically and so not 100% accurate during the live event, but they will be edited so that they are correct on the event recording. After the event, we will send you a link to the event recording in which you'll be able to watch on our website or on our YouTube channel. This is also where you'll be able to find all the previous event recordings like this. If you would like to ask a question during today's set discussion, you'll need to click the Q&A button at the bottom of your screen, which will bring up the box where you can type your question. We know today's topic is of great interest and we will do our best to answer as many questions as possible. We aren't able to answer any personal medical questions during the event, however. We're now going to ask you some poll questions so that we can find out a little bit more about you today. And these should pop up on your screen now. And they ask about your reasons for attending today, how you rate your knowledge of dementia research, and if you've attended one of our online events about research before. So I'll just pause a second to give you a chance to fill those in. So we know people often attend these events because they have personal experience of dementia within their family. So that's coming out with quite a few answers. People's work is related to dementia as well. We have people here who have dementia themselves or they're caring for somebody with dementia. And also people who don't have a personal connection but want to find out more. We welcome everybody. These events are a great way to find out more about the research that's taking place. Lovely. And we've got a bit of a split. So some people have attended some of these events before, so it's wonderful to have you back. And for others, it is the first time you've attended an event about dementia research. So I hope you enjoy it and you're able to take on board lots of information today. Lovely. So thank you. So I think we'll move on now. So as I said, we have a mix of first-time returning attendees. So welcome to you all. Just for those of you who are new to these events, I'll briefly introduce Alzheimer's Research UK. So we are the UK's leading dementia research charity, and we're working to revolutionise the way that we treat, diagnose and prevent dementia by investing in the best research, powering the most forward-thinking scientists and joining forces with world-class organisations. But all of this is only possible thanks to our amazing supporters who donate and fundraise, volunteer and raise awareness of our work, plus so much more. And it's with your support that we will keep going until every person is free from the heartbreak of dementia. And so today I just wanted to highlight one way that you can support us and get involved over the summer, which is our Walk for a Cure events. And there are 6 family-friendly and accessible 5-kilometre walks, which are taking place in Leeds, London, Poole, Birmingham, Cardiff and Edinburgh across May, June and July. And then in September, we have 2 multi-distance hike events in the Peak District and in the Cotswolds, where you can pick your distance from 6 kilometres all the way up to a challenging 42-kilometre walk. And these events are all a fantastic way to come together with friends and family, and every step brings us closer to a cure. And if you don't fancy walking but would like to support these events, you can also sign up to volunteer and support us on event day. So check out the website for all the details and where you can sign up. But back to today's event. My team put on events like this so that you can hear directly from Alzheimer's Research UK funded scientists as they share and discuss their research and how it could change lives. So without further ado, it's my pleasure to introduce our first guest today, which is Dr. Emma Murphy, who should join me on screen now. Lovely. Emma, can you give us a brief introduction?
Hello, I'm Dr. Emma Murphy. I'm the head of biology at the Alzheimer's Research UK funded Oxford Drug Discovery Institute, which is part of the Drug Discovery Alliance. And there are institutes at UCL in London and also in Cambridge. We really exist to bridge the gap between academic research into dementia-causing diseases and the pharmaceutical industry, who can take this research and make new treatments for dementias. We use a variety of sources to identify things that we can modulate to which we hope will be able to treat dementias. And a lot of our targets come from genetic research. So comparing the genetics of people who go on to develop dementia and those who don't, and seeing what those differences are. We really are the earliest stage of drug discovery. So we take the basic academic research that's being carried out and we try to find small molecules that can modulate these targets. We also try to validate these targets as potential therapeutic targets. And we do that using cellular models of Alzheimer's disease that we develop ourselves. So we take drugs from the very, very earliest stages up to the preclinical stage. And that's where a lot more funding and a lot more work is needed for these. So then we pass these on.
Thank you, Emma. And I'd like to introduce now Dr. Joyce Huang, who's our second speaker today. Hi, Joyce. Do you want to just introduce yourself briefly?
Yes. Hello, everyone. I'm Joyce Huang. I'm the lecturer in pharmacy practice at the University of Manchester. I'm the pharmacist by the background, but I have been working on the drug utilization research for several years. My main research area focuses on aging populations, comorbidities, and polypharmacy. So, of course, the dementia population is one of my big research area. So in recent years, I'm very keen to incorporate the more advanced health data science skills into the drug utilization research so that we could potentially answer some research questions that we cannot observe or find the answers in the clinical trials. So now I'm leading on a pilot project funded by the Alzheimer Research UK to investigate the potential effect of one of the anti-diabetic medications on preventing or delaying vascular dementia in people living with type 2 diabetes.
Lovely, thank you. So kicking off with our first question, Emma, I'll come to you. So you said a bit about, where you start and where you take your research up to, but could you talk about, who's making up the teams that you're working with and how do they kind of come together in this unique way to take those ideas from the early stage up to the point where they could be developed by pharmaceutical companies?
So at the Oxford Drug Discovery Institute, we have a huge team, and our team is kind of split into three different types of scientists. So once we've got our target, which mainly comes from collaborators, literature, data that's already out there, we have an assay development and screening team. So this team is a team of scientists that can look at ways of modulating targets. So they find specific readouts of how that target is being modulated. We also have a target validation, a more biology type team, and they're the ones that build our cellular models. So we take stem cells, human stem cells, and we reprogram them into different types of brain cells. So neurons, but also some of the immune cells in the brain, so microglia and astrocytes as well. And we build models of different tissue cultures, and then we can use those to test compounds to see if they are potential therapeutics. We also have a large medicinal chemistry team. So this team are able to take the compounds, which might not be very drug-like, and change some of the molecules to make them more drug-like. So things like increasing their chances of getting into the brain.
Lovely. Thank you, Emma. Joyce, coming to you. And the first question I was going to ask you is actually, Peter has just asked it as well. So when you're looking for medicines that you could repurpose, how do you find them? So how did you first decide to select this diabetes medicine as a potential thing to look at for dementia?
Yeah, so maybe I probably need to explain the terminology first. So we call the epidemiological research. So let's say it's different from the traditional clinical trial because in this type of research, we will use the existing database and to look into any drug issues that we cannot observe in the clinical trial. So let's reframe this very complex word into the population-based study, okay? So some of the population-based studies has found that the TZD, one of the anti-diabetic medication, actually is related to the lower risk of dementia, including the vascular dementia. And in some, although the underlying mechanisms hasn't been clear, but there are some previous studies identified that, okay, the TZD may be able to improve the cerebral brain damage and also reduce the inflammation in animals models, which might be related to the onset or the progression of the vascular dementia. So that's why we selected these drugs. Yeah.
Lovely. Thank you. Em, I'm just going to come back to you now. So let's just break down, I guess, some of the terminology of drug discovery. So when we say target, what do we mean by target? And when you talk about modulating it, what does that actually look like?
So when we look at the genome-wide studies, so when we compare people who are likely to develop dementia or people who have developed dementia with people who haven't gone on to develop dementia, there are often genes which contain mutations that have gone wrong in the people who have gone on to develop dementia. So that would be a risk factor. And these aren't, it's not like a inheritable condition. It's not like this gene's gone wrong, you will get Alzheimer's. It's more of a risk factor. So if you've got a lot of these different genes, you're more likely to go on to develop dementia. So when we found a gene, that is what we would call a target. And say, for example, it's something that's more active in people who are likely to develop dementia, then we might want to modulate it by inhibiting it, so reduce its activity. And we would want to do that using a small molecule. So we run large high throughput screens, so we can test whether a small molecule can reduce the activity of that target. And we can test hundreds of thousands of small molecules in a day. And that's how we do our high throughput screening.
Fab. And so small molecules, they're essentially just little bits of chemistry, aren't they, that you think could attach onto this gene or this protein, that the target you're trying. So it's basically like a lock and a key sort of mechanism, isn't it?
Absolutely. Yes, that's exactly it.
Wonderful. Kind of thinking about how the world is changing, you know, AI has come in, it's revolutionizing a lot of the work that scientists are able to do. I'll come to you in turn, but like I'm really interested to understand how this maybe has benefits for speeding up aspects of your research or helping you to identify, you know, things to look at. So I wonder, Joyce, if I come to you, you know, does AI mean it's easier to identify some of these drugs for other conditions that might have benefit in dementia?
The simple answer would be yes. So if you were in the Alzheimer Research UK conference last month, then I think there's a research team from the Queensbury University. Actually, they try to use the machine learning algorithm to identify some metabolites or biomarkers that could be causally related to the onset of Alzheimer's disease. So of course, I would say it would speed up in some ways. So the AI, the full name is artificial intelligence. And actually behind of the scene, behind of the AI, actually it's just multiple algorithms, they kind of through the different process, etc. But I just want to say that utilizing this kind of high technique, aka the machine learning algorithm, I think all the people need to be cautious about that because there's quite huge criticism of artificial intelligence. It's just that within the machine learning algorithm, it's just like the black box. So just that if we threw out any, we can throw any data, any gene, any information into this algorithm, and we can get something in the end. But sometimes people don't understand 100% what has happened within that algorithm. So it called the black box mystery. So yeah, but I'm more kind of optimistic to that. Yeah, I think that we can overcome some challenges and hurdles. Yeah.
So it allows you to look at more
Yeah.
data more quickly, but you
Yeah.
still need to add a little bit of kind of human critique and thinking to the results that you're getting out. Yeah, really, really good point. Emma, coming to you. So does it help identifying more of these kind of little biological things that you can try and target?
Absolutely. So I think AI is being used in Alzheimer's research in every single aspect of it. And one of the places where we use AI is to try and help us identify targets. And that's because the machines can learn and analyze data a lot quicker than we can. And it's also able to spot patterns that maybe we as humans wouldn't necessarily spot. And as Joyce said, we do then validate these things. We don't just take the computer's word for it. We check to see if they are good targets. But we absolutely use it, and I know that it is being used more broadly in things like identifying people who are likely to go on to develop dementia, which will help greatly in selecting participants for clinical trials, especially pre-symptomatic people, which is the people we really want to treat. So I think it is going to be beneficial, and it's definitely the future.
Fab, thank you. So you've mentioned, Emma, small molecules quite a bit, and I wonder if we could just talk a little bit about why small molecules, what's useful about them. So does it relate to the fact that actually it's quite hard to get drugs into the brain because it's protected? And is that the particular bit of chemistry that's most likely to get in?
Absolutely. So the brain is protected by the blood-brain barrier. And this is just to stop toxins and pathogens entering into the very delicate ecosystem that's the brain. So large molecules, things like antibodies, which we produce in the periphery and the bloodstream, can't readily access the brain. And so these small pieces of chemistry really are only a few molecules, and they do find it easier to access the brain. Not all of them do. There are lots of drugs out there that aren't brain penetrant. They can't get across that barrier. But there are techniques and ways to develop chemistry that is able to cross that barrier. And even larger molecules now, things like the monoclonal antibodies, people are looking at tagging them in a specific way so they can be carried across the barrier by a process called receptor-mediated transcytosis, which is a bit like a Trojan horse. They sneak across the barrier whilst attached to something that is supposed to cross the barrier. So it's a challenge in CNS research, but it's definitely something that we're starting to really think about overcoming.
Lovely. Yeah, that I think is one of the big challenges, hasn't it been, for developing these treatments, is just simply getting them through and understanding bits of biology, like you said, the Trojan horse, where you can use existing mechanisms to get these drugs in is really important. Thinking again of some of the challenges of developing drugs for dementia, Joyce, I wonder if I'd like to come to you. So with your pharmacy background, you've talked about the fact that people are often taking multiple medications. So what challenge does this present when we're trying to find new treatments for dementia?
So first of all, I think people tend to be older or have more complex comorbidities. They are usually excluded from clinical trial because other coexisting diseases or other concurrent medications could be the confounder. Okay, the interpretation of confounder is kind of interfering factors in our research. So it will make the clinical trials result not causality enough, which is so I think that is not what pharmaceutical companies want, because they want to prove the causal effect of the drug A so that they can get the drug license approved. So unfortunately, I think most of the older people or with comorbidities, for example, living with stroke or living with type 2 diabetes, they are not eligible to be recruited in the clinical trial. So I think that is really challenging, that we want to use the traditional drug development pathway to find the dedicated treatment for this specific population. Yeah.
Yeah, so it's understanding that whilst you're doing the trials and it might be people who don't have other conditions, actually in the real world, the vast majority of people that we want to get these drugs to are likely to have other things going on, and just being able to account for that. But then, as you say, your research is looking at a drug for another condition that can also have benefits. So it shows that having that bigger viewpoint has some benefits, because you could spot other patterns going on. Yes. I think we had some questions come through ahead of time that were asking about, what types of dementia are we developing drugs for? So we know last year there were 2 new drugs licensed specifically for Alzheimer's disease, but Alzheimer's is not the only form of dementia. We want to be developing drugs for all the different types of dementia that are out there. So I wonder, Emma, how do you at the Drug Discovery Institute and the wider Drug Discovery Alliance, how do you take on that challenge of developing treatments for as many types of dementia as possible?
So we look at all forms of dementia. We have a focus on Alzheimer's because it is the leading cause of dementia, but we do look at some of the rarer forms as well. And one of the things about the rarer forms, mechanistically, they are quite similar to Alzheimer's. So there are pathways and different types of biology going on in the brain that, if you ameliorate it in one form of dementia, is likely to be beneficial in other forms of dementia. So we don't solely focus on Alzheimer's, but we do look at rarer forms of dementia, things like tauopathies, the primary tauopathies. We've got a large program that's co-funded by the Rainwater Charitable Trust, which is specifically designed to look at these rarer forms of dementias. So it is something that we consider, and actually I think mechanistically it's very informative for us to look at these other forms of dementias.
Thank you, Emma. And Joyce, the treatment that you're looking at specifically for vascular dementia, but given we know that, heart health and blood vessel health underpins probably all types of dementia, is it that whilst you might look at initially in vascular dementia, that actually it could have benefits for other types of dementia as well?
I'm not quite sure at the moment. Yeah. So yeah, because I'm not a lab worker, but from the data perspective, I think all the population-based studies still focus on Alzheimer. Is still focused on Alzheimer disease, and I think the biggest reason is because they can get the enough sample size from the existing database. Yeah, but however, I think there's ongoing clinical trial on another drug repurposing issued by another pharmaceutical company. I think they are also investigating if another type of anti-diabetic medications is beneficial for the vascular dementia. So let's see how things going. Yeah.
Lovely, thank you. Okay, just having a look through at some of the questions coming through. I think this is probably one for Emma. So you mentioned high throughput screening and like how you can look at lots of molecules at the same time. How many can you look at in kind of one experiment at a time when you're doing something like high throughput? Are we talking tens, hundreds, thousands?
It all depends on the readout we're looking at. So if we have a target, say it's a small molecule in the brain, an enzyme in the brain, which is a protein that has an activity, and we're just looking at that activity, we can often do that in a biochemical format. So we purify that protein, we can make it fully lab made, just pure in-house made. We don't need humans or anything for it. And then we just have that protein and then the thing that it acts on, which is called a substrate. And so that might be something that when the enzyme, the protein, our target has acted on it, then becomes fluorescent or changes color. And we can measure that event. If we have something that's really that simple and all we want to do is prevent that our target from forming that color, then we can test 1,536 compounds on one plate, which will take 1 minute. So we can test hundreds of thousands of compounds a day in that case. Sometimes we don't have the luxury of having that target that available and knowing the substrate and things like that, in which case we need to use wholesale systems. But even then, we usually do that in a 384 well plate format. So we can look at 384 wells with live cells growing and monitor changes that we think are beneficial for the cell's health. And so if that particular well has our small molecule, our drug in it, and it's beneficial to health, we can see that. So high throughput really is quite quick. And we're very lucky to have the equipment that we need to run those kinds of screens.
Yeah, I think I visited once before, and you have these kind of robots, and they can use sound waves and really incredible technology to actually speed up and make sure these experiments are really, really precise. So yeah, it's a really fantastic kind of environment to see happening. Apologies, I only ever seem to get a cough when I'm doing something that's, public like this, so bear with me. We've had some other questions actually about, I would say the theme is international collaboration. And I wonder if you could both reflect on this from both of your areas. So, the world, we live in a digital world, we're much more connected than we used to be. But what does that look like kind of day to day in research? Is it the access to databases from around the world? Is it the ability to collaborate with people in different nations? How do we in the UK link up with research in the rest of the world? I don't know who would like to go first on that one.
We can.
Yeah, off you go, Emma.
Yeah. We actually have a really, really collaborative program here. So we collaborate with people all over the world. And that is things like sharing databases, but also we have projects that other people in the US and all over the globe are working on the same project. So we might do, say, the high throughput screening part of it, because we have the technical capabilities to do that. And then they might take our small molecules and put them in something that's a bit more disease relevant. So a model or cell-based system that is mimicking a bit more the natural brain environment. So we send samples, we have regular meetings, Teams meetings, occasionally in-person meetings if someone happens to be in whichever country at the time. But we are very collaborative globally, and I think that's how it has to be. This really has to be a joint effort.
Thank you. Joyce, from your perspective.
Yeah, probably as the data analyst, we are less lucky than Emma's experience because the data access is always very challenging due to the very strict data protection regulation across the UK or including in the UK. So I think it's possible that we find the research collaboration globally from using the database from different countries. However, we probably need to replicate the studies using the different existing database instead of merging all the data in one pool and do the one study at all. Because in my head, I think it's unlikely to happen according to the current regulations. But yeah, if people are interested in some prediction model or some very complex machine learning algorithm, usually when we developed one model, we will use for example, the database in the UK, and then we will validate externally using the different databases, for example, from different countries or with the entirely different patient characteristics. And then so that we can confirm that prediction model will both internally and externally. So I think, of course, research collaboration is very important, but data access is always our first thing need to overcome. Yeah.
Lovely, thank you. Thinking still of the kind of the bigger picture of the drug discovery and development landscape. With the exciting news last year with the 2 new licenses for treatments, have you felt an impact of that on your work? Does there feel like there's a different mood in the kind of research space? Is there, more funding coming in from pharmaceutical partners? Emma, I don't know if you want to give us a start on that.
Absolutely. I think that they're monoclonal antibodies to A beta that were FDA approved. And I think that really has reignited the pharmaceutical industry to start looking back into this issue again. There were decades where lots of things were tried and nothing seemed to be working, but now we really have got something that is starting to show progress. So although I think obviously there's a lot more research that needs to be done, these are first generation, there are issues, but people are looking at that now. And I just think that it really has brought people back to neurodegeneration research. So it's really just kick-started it, which I think is amazing.
Thank you. Joyce, do you have anything to add on that? Has the mood shifted, do you think, in trying to find new treatments for dementia with the positive news from last year?
I'm personally quite excited about that because it's the first drug that can slow down the early stage of Alzheimer's disease. But because it hasn't been available in NHS, so it also kind of highlight the challenges between the new drug availability and also the healthcare systems issue. Yeah.
Thank you. Just kind of weaving a few questions together that we've had come through live today. So Joyce, you mentioned we were talking a bit about the challenge of people having multiple conditions and being on multiple medications. And when we're thinking of this, are there particular ages or points of people's lives where you start to see? So, what sort of age might you see that people are on more medications and therefore it becomes more complex? And also, you mentioned some of those different conditions like diabetes and stroke. Are those the main ones, or are there kind of other conditions that often appear alongside dementia as well?
Okay, so I think using the very specific age cutoff point probably is a bit awkward because if you so let's say if people they are staying very physically active until their age 65, I think their condition is still pretty good. So I would say it's closely related to lifestyle. So let's say obesity, sleeping problem, drinking, smoking, this kind of thing. And also the physical activity is really important. But in general, I think people age more than 65 or 7, they tend to have more than 2 coexisting conditions, to be honest with you. And also they would have the higher percentage of polypharmacy. Polypharmacy is actually not a bad thing. It's just that we need to make sure all the drugs you are taking at the moment meet what you need. So they just need to be reviewed on a regular basis instead of that, because I'm taking this drug probably since 20 years ago, so I just need to take this right now. It doesn't mean that. And I think for dementia population, there's another tricky point for the healthcare professionals is actually the prescribing has been recommended for the people living with dementia because the prescribing has been found to improve their health outcomes. However, the prescribing is not that easy, right? Because you will need to get your medications up to date, get everything right. So I just want to let everyone know that actually we have the structured medication review, that is policy in our primary care system in the UK. So if you are wondering that whether you still need this medication or not, you can just speak to your GP or clinical pharmacist for their for their opinion. Yeah.
Yeah, to get that review to make sure that everything that you're taking is still needed for you right now. Wonderful. Emma, I'm going to come back to you, and this might be a bit of a tricky question, but we know the drugs that were licensed last year, you've mentioned they were what we call monoclonal antibodies, and they are given to people in an infusion. So they have a kind of line in their arm that then brings the medicine into their body. But we wouldn't say that was probably the ideal, accessible way of giving a treatment. So are those aspects of, how a drug can be given, are they considerations that you can make at this really early stage? So, for example, small molecules, how would those get into the body if they were actually turned into medicines?
So, excuse me. So small molecules would more likely be orally available or through a simple intravenous rather than the infusion. So the routes of administration for small molecules are generally better than they are for these large monoclonal antibodies. But the monoclonal antibodies themselves, at the minute, they're infusions, but a lot of work is being carried out into improving that route of administration. And I think that will really help with some of the challenges of prescribing these and getting these going in countries where they haven't been fully approved or accepted on the healthcare systems. So I think that these are considerations that pharmaceutical companies make. They know that it's tricky to do these infusions, and it's a process that people living with a diagnosis of dementia aren't going to want to go through on a regular basis. So that it's a continuous effort to improve the method for delivery. But a small molecule that was orally bioavailable, so you take it, it gets to the brain, would be, the gold standard. That's what we're really aiming for.
Because then, they can be prescribed, people can take them home, and they're not required to have these regular visits, and it's a lot less difficult and much more accessible for them to get it. Yes,
exactly.
That's the goal. And thinking about the research you're doing, the million-dollar question that everybody wants to know is how long does it take? So could you give us an example, Emma? You've just started a project in the Drug Discovery Institute. You've got, some early data that shows that this particular aspect of biology could be a target, could be druggable. How long, if everything went well, could it take from that moment to it potentially being in a pharmacy, being given to people?
That's a really, really challenging question. It is a process, because even if we found the right molecule, we then need to develop it into being more drug-like. We need it to be potent. We need it to be selective. So sometimes we'll find something that is able to modulate our target, but then if we look at other targets, it modulates them as well. And so there are lots and lots of different steps we need to do to ensure that not only is it efficacious, not only does it work, but also that it's safe. And so, unfortunately, it just takes time. You can't fast track these things. One of the key things about these newer drugs being approved is that it has put in place some of those regulatory mechanisms. So once the actual research is done, hopefully the red tape and the process of getting them approved will be a bit quicker. And also things like we're always identifying biomarkers. So these are small biological molecules that you can find in people's blood or tissues or easily accessible samples that you can correlate to disease and the level of disease and use it as a way of telling whether something is working in the body. So when we have those in place, it means that we can do a lot of the clinical studies much quicker, because we know, if we don't see that anymore, then we're doing something beneficial, or if we've decreased it, this is beneficial. So while we're working on the drug discovery process itself, other people are working on finding the ways that we can monitor how well that drug is working. So I'm sorry, that wasn't a very concise answer. It wasn't. I didn't give you a specific time frame, but
we're talking decades, though, aren't we? Yeah,
we are talking decades. I think best case scenario from our lab to the pharmacy, 15 years, 12 years.
Yeah.
It's
So that's because
a process.
it needs, as you say, it needs to be effective, but it also needs to be safe and try and minimize side effects and ensure that it's really doing what we want it to do. Fabulous.
Yeah.
Well, we are rapidly running out of time, so I'm going to come to my last question. And I always like to look for the grains of hope, the positive things that are coming from research. So I'll come to each of you in turn. What is it that's currently happening within your areas of research that's most exciting you for the future? Who would like to go first? Emma,
Okay.
off you go.
So, dementia research is coming along so quickly now. In the last decade, there have been so many new discoveries, so many new types of biology that are involved have been kind of elucidated. It's a bit like a jigsaw, you know. At first, people were finding the corner pieces, and now we've got the side pieces, and we're filling it in now, and it's just getting quicker and quicker and quicker, and we're building up our understanding. One of the things that is quite new that I think is really exciting is that these genome-wide association studies have shown, you know, the things that have kind of gone wrong, and so the people who are more likely to develop dementia have these certain things, and the people who don't haven't got them. But it's also found what's known as resilience genes. So these are things that people can have a lot of pathology, so they can have the toxic protein plaques, the amyloid plaques, but cognitively their cognition is really good. So identifying the things that they have, which people who do go on to develop dementia don't have, is also another source of potential therapeutics. So it's a really exciting area that is gaining momentum at the minute.
So I guess those are ways that could protect the brain as well as trying to slow down some of the damaging processes that are happening. Yeah, that is very exciting. Joyce, what's hopeful? Where's the hope coming from for you?
So, so I think for the ongoing research, I think I'm particularly, in addition to my pilot project, I'm particularly interested in the another drug repurposing project. We are talking about this semaglutide, this GLP-1 agonist, to see if that have potential benefit for Alzheimer's disease. And for future hope, okay, I think we still have a long way to go, but I would like to build up the personalized treatment prediction model if possible. Yeah, but as I said, there's a different multiple steps and there's different challenges we need to think of later. Yeah.
Lovely. Yes, we did have some questions about personalized treatments, and yeah, that's definitely a big future hope for many of us. So we're almost out of time, so I'll say goodbye to our speakers now, but if everybody holds on, because there's just a few more things. So thank you so much to Joyce and Emma for joining us today. And just got a final few things to run through. So firstly, we just want to do a quick poll to get some initial feedback on today's event. So that helps us to understand what's been going well, where we can improve. And we'll also be sending a more detailed feedback survey to you later today. So please do share any comments with us as they help us to understand what's working well and where we can make improvements. So yeah, the poll's on the screen now. And we want to know, you know, what was the content all right? Were we too technical? Did we get it about right? Would you recommend these to friends and family? And do you feel like you learned something today? So those are just some really useful indicators as to how the event's gone. So I'll just pause a second while people fill that in. But it's looking pretty positive. Got it about right. Some kind of technical language that I hope we can break down a bit more. Most people agreeing that they would kind of recommend, and they do think they learned something today, which is fantastic. And it kind of gives us the reason to keep doing these events for you as well. So just a reminder then, we shared a bit about our Walk for a Cure events that are happening this spring and summer from May to September. And as I said, there are a range of locations and distances that you can pick from, and they're a really wonderful way to come together with friends and family and to walk together to bring us a step closer to a cure for dementia. So if you are interested in finding out more, do check out the website where you can find out more and sign up. And I know we had quite a few questions come through today that we weren't able to answer, or somewhere you might need more personalised information or signposting. And our dementia research info line is here to help. The team can answer questions and signpost on to other organisations who can offer support. And they can also signpost you to join Dementia Research, which is a service that can match you to research studies. And anyone over the age of 18 can register to be in Join Dementia Research. You do not need a dementia diagnosis to take part in studies. And you can find out more on the website or by calling our Dementia Research info line. And we'll be putting details in the chat and also in the follow-up emails for how you can sign up for that. We're busy planning our next event, which will take place at 1 p.m. on Tuesday, the 10th of June. And what we're talking about is how right now doctors can't really predict what someone's future may look like after a dementia diagnosis, such as how fast their symptoms may progress, and that can limit how individuals and families can prepare for the future. And so in June, we'll be joined by two scientists whose work aims to change this by studying how dementia progresses over time and identifying better ways to predict an individual's journey after diagnosis. I really hope you can join us again then as we explore that fascinating and vital area of research. So all that leaves is for me to say thank you so much for coming along today and taking the time to find out more about research, working to create new dementia treatments. You've heard about the exciting and important work that's taking place and some of the challenges that the researchers are trying to overcome to make this progress. And thank you for all your support that has got us to this point. And if you're interested in other ways to support the work of Alzheimer's Research UK, check out our website for lots of ways to get involved. Thank you again for joining us today. I hope you found it useful and we hope you can join us in June for our next event. Thank you and goodbye.